The genetic information in our genome is silent. It needs to be given voice. The RNA polymerase is the central enzyme in the cell nucleus that brings life to our genes.
Going from DNA to RNA has been a complex gene transcription process, one already identified back in the 60s, but which has still not unveiled all its secrets. Here in Göttingen, Germany, Patrick Kramer, a chemist by training, is working on RNA polymerase, a key complex for life in all organisms.
The RNA polymerase will move along the DNA and it will copy the gene, and the copy that it is producing is the RNA molecule, the messenger RNA molecule. And this messenger RNA molecule can be used to synthesize proteins in a second step. And the proteins will do whatever they do. They have all these different functions that then bring about life.
To better understand this molecular machinery, Patrick Kramer and his team were able to reveal the tri-dimensional structure of RNA polymerase. The discovery was made possible thanks to electron microscopy, a technique by which the RNA polymerase could be viewed firstly in 2 dimensions and then visualized in 3 dimensions.
These are the single particles of the RNA polymerase, and these are the 2-dimensional images that we observe for these protein complexes. And once we collect many of those images, we can then process the data so that we can reconstruct a 3-dimensional volume for our protein complex.
Patrick Kramer also used other technologies such as crystallography, used here in the Max Planck Institute for Biophysical Chemistry in Göttingen, for creating this video. Researchers, scientists and students can see the RNA polymerase as it functions in our cells.
So visualizing structures of proteins or protein complexes really makes it very easy to understand how things are working in the cell.
If you just have a 2-dimensional picture, you have no idea what's actually happening in the cell and what molecular mechanisms are driving these processes. So I think to be able to see that with such precision is incredibly important for us to understand what's going on.
And with the structure, you can make predictions. Some ideas don't come anymore because you immediately see that this is entirely impossible. And the others come because there are other possibilities which you couldn't imagine just based on 2D structures.
Seeing RNA polymerase in 3 dimensions isn't enough for explaining the whole cellular transcription process. There are other elements, and those are the ones Patrick Kramer's teams are studying today.
The RNA polymerase is like the main worker in all of this process of transcribing the DNA into RNA, but there's a lot of different molecules involved.
This is a pre-initiation complex that Patrick works on it for the last 2 decades. He majorly resolves the left half, which contains mainly the polymerase itself, and there are several initiation factors that involved in this process to help the polymerase start on the DNA template.
Understanding the RNA polymerase structure and environment so as to better explain how it functions. Using functional genomics, Patrick Kramer aims to take his research further in our understanding of living beings.
Functional genomics is asking questions to living systems, and it allows you to extract information from living cells. So now we can use these methods in order to study gene transcription in the natural context within living cells. And our dream is to be able to transcribe an entire genome in the test tube. Because if we can mimic the situation in a living cell, then we can understand much better how it really works.
You need the knowledge so that you can understand what is wrong in different diseases, and it can also be interesting for targeting new therapies and so on, because if you know the whole mechanism, you know what is exactly happening in different steps.
By controlling the development of illnesses such as cancer, the research conducted here takes knowledge forward. Patrick Kramer was recently able to apply his expertise to describing the SARS-CoV-2 polymerase, thus opening up the way for more research and action against COVID. A plunge into today's world of medicine, but not one that will prevent Patrick Kramer from being firmly anchored in his real universe, fundamental research.
It's very important to have those people like Patrick who just go after something that is unexpected. If you go after something fundamental, if you're driven by the desire to understand something at a basic level, then in the end it's likely that you're going to make breakthroughs because this is where science makes big leaps.
At my heart of hearts, I'm still, somebody who is in basic research. But we are open towards potential applications, and we would like our knowledge to be used in order to find applications. This could add societal value to our work so that we don't remain at the level of basic research, but we also offer that knowledge so that we can help society.